Welcome to this BLI LAMP seminar to everybody, and especially to our speaker today, who is Dan Cojoc, I should say. I always say Cojoc, it's not fair. He is at the CNR Italy. He got his university degree at Bucharest, and he has worked in biophysics for quite a long time now. I see him at conferences occasionally, and I see his talks, and every time I see his talks, it makes me feel like I wish I had done that. And so that probably is a mark of a good researcher. If you feel like all the time the person is doing something better than you are, it probably means that he's good. So he's done a lot of work, particularly on optical tweezers, digital holographic microscopy. He spent a lot of time working on light interactions with neurons and various cell types. He has published more than 130 papers, has 3 patents, 11 book chapters, and is a reviewer for 10 scientific journals. And he is an adjunct professor at the University of Trieste, which is in northern Italy. And so I'd like to welcome him to this BLI talk, to this LAMP seminar, and cordially hand it over to you, Dr. Cojoc.
Okay, thank you very much for very nice introduction, even if it is a bit overestimated on my research activity, but it is welcome anyway.
No, it's not.
And thank you very much for the opportunity to present my work, part of my work within the Virtual LAMP seminar series. And I'm really honored to do this. Today I will speak about cell mechanics by light. I will begin with a short introduction for the place I am from. So Trieste, close to Venice, and close also to Austria, Slovenia, Croatia, and other countries. So it is in the middle of Europe, let us say. Our institute, the headquarters actually, is in Area Science Park, which is placed at about 10 kilometers from downtown. And here there are 2 main facilities, Italian Electra Synchrotron and Fermi Free Electron Laser is a more recent facility. And many of my colleagues work as scientists, beam scientists, or responsible for Electra Synchrotron. So they run experiments, they keep the instrumentation, develop the instrumentation there. Also, I was actually, when I came to Trieste, I was involved in developing diffractive optical elements for X-ray microscopy. So those were my first two years. Then I understood it is too difficult, so I moved to optical microscopy. It's a joke, of course. Okay, so we have also other branches in Perugia, University of Perugia, in Cagliari, in Sardinia, and in Genoa, and one in France at the European Synchrotron Radiation Facility. Briefly, my institute is an interdisciplinary research institute belonging to the National Research Council, or Consiglio Nazionale delle Ricerche, CNR, that carries out, our institute carries out research in the fields of condensed matter physics, nanoscience, and biophysics. And the mission is twofold. One, to gain fundamental knowledge at the nano and micro scale that enables novel technologies, and to develop advanced methods for the synthesis, characterization, and modeling of functional materials and biosystems. The mission areas are 4: energy and environment, quantum materials, sensors and devices, and biomed, which is quite young area. And my lab and many other labs are part of this area. If you are interested, you want to visit for more information, please visit the website of our institute. Now, if we begin speaking about cell mechanics, so it is already known, even if it is not very recent, that cells generate and sustain mechanical forces within their environment as part of their normal physiology. So besides the biochemical processes which characterize the cell behavior in a physiological conditions. The cells respond to exogenous and endogenous mechanical stimuli. For instance, exogenous is the response of the epithelial cells due to the flow shear of the blood in the vasculature. And endogenous arises from cell matrix and cell-cell interaction. And probably the most studied property of the cell, mechanical property of the cell, is the stiffness, but also in correlation with the stiffness of the environment. And it is known that the cells respond to the stiffness of their environment by changing morphology, motility, proliferation, or by altering their differentiation state. Not only the cell changes the morphology and other properties, but also the cell changes the substrate properties as a response to the mechanical stimulation, as it is shown in this figure for fibroblasts, which once it senses the substrate, it changes mechanical property, changes morphology. Yes, for instance, it enhances stress fiber formation, the contractility, but as a result of this process, also the cell changes the matrix deposition and the reorganization and alignment. Even if this is not a new observation or new achievement, because it was first reported, the interaction between the fibroblast and the substrate, which changes stiffness, already in 81 by Harris and colleagues in a very beautiful work. Actually, there are many other works. I want just to cite the findings, some findings reported in this work. First, the strength of cellular traction differs greatly between cell types, and this traction is paradoxically weakest in the most mobile and invasive cells, as leukocytes and nerve growth cones. Fibroblast exerts forces very much larger than those actually needed for locomotion. This strong traction distorts collagen gels dramatically, creating patterns similar to tendons and organ capsules. So this was a source of research, and many findings already implemented or transported in medicine, in real medicine. And the morphogenic rearrangement of extracellular matrices is a primary function of the fibroblast traction, and it explains its excessive strength. So actually, reading this paper and the other papers at that time in 81, we realized that with much less technology from the point of view of experimentation, people already observe and drive very useful conclusions and on cell mechanics. But still, there are many questions to be answered. And of course, we can take advantage of the new technologies. I also, if there are young people or people interested, I also recommend the website of Professor Harris' lab, where he deposited the movies of the behavior of the cells in different conditions for the substrate. They are wonderful. I mean, they are old. They are not very fancy as quality, as colors, but they are wonderful as message and as information seeking. Cells are not passive materials, they are active materials that can detect mechanical stimulation by the activation of mechanosensitive signaling pathways and respond to physical cues through cytoskeletal reorganization, chemical signal activation, and force generation, among others. I think these are the most important. I took an example from cytotoxic T cells. And from this paper by Basu all in Cell 2016, where they show that CTL uses mechanical force to potentiate target cell killing. Cell killing is obtained by when CTL secretes toxic proteases and the pore-forming protein perforin into the synapse. And what they did, they induced an increase of the tension of the membrane tension of the at the immunologic synapses, and observed that in this way the perforin activity was potentiated. And well, this happens of course spatially and temporally, and the nice thing is that this conclusion that cells use mechanical forces to control the activity of outgoing chemical signals can be generalized, not in the same way, but can be source of inspiration, let us say, for research for many, many other type of cells. And still another example, I do not enter into detail, is the emerging role of mechanical forces in epigenetic regulation and chromatin remodeling. I just indicate two, in my opinion, good reviews on this. Now, if cells have mechanical properties, these mechanical properties can be changed, and the mechanical behavior can be changed by genetic mutations, by pathogens, or by interaction with the environment. This means that cell mechanical properties and cell mechanical behavior can become a hallmark and symptom of variety of pathologies, highly motivating the research in the field. And since signaling pathways elicited by mechanical stimuli begin to be more and more elucidated, and mechanobiology has the potential to reveal new targets for diagnosis and therapy. So which properties, some properties, mechanical properties of the cell, morphological, shape, volume, size, area, elasticity, viscosity, fragility, adhesion, motility. So they are quite complex properties. Now, if one thinks to measure, how to measure, to evaluate the mechanical properties, should design methods, techniques to do this, and should take into account the complexity of the cell, behavior in different conditions, of the cell type, of the cell state. So, and also the difference between the experimental methods and the theoretical models with which we try to understand those results. There are many techniques for cell mechanics study, mainly for cancer cell. I report here some taken from a review by Suresh. They are with probes. So in these techniques, they are used probes. For instance, in AFM, the tip is a probe, and the mechanical contact between the probe and the cell. For AFM, for magnetic tweezers, for cytoindentation, for microplate stretcher, for micropipette aspiration, for microfluidic assay, also for the laser optical tweezers, even if this is not the only experiment possible to do stretch of the cell, as we will see. So mechanical contact also here, the tracking force microscopy, when the cell is placed on a substrate which can be deformed by the cell, and by optical microscopy we follow this deformation, or we can inject nanoparticles in the cytoplasm and track the nanoparticles and deduce the viscoelasticity of the cytoplasm, or by shear flow, which mimics the flow of the blood flow in vasculature, for instance. And this is the optical structure in which light is used. So we do not have mechanical contact for the deformation of the cell. I think it is the only method without contact, mechanical contact, reported here. So for local mechanical investigation, I would say that the most used, or the most used are AFM, optical tweezers, mechanical tweezers, and nanorheology. But there are also other methods, optical methods. Quantitative phase imaging, which involves the measurement of the phase retardation of the light passing through the sample. And since the phase cannot be detected directly, it is detected through interference, so obtaining what is called the hologram. And then reconstructing the hologram, one gets the height of the cell, the volume of the cell, the refractive index, the dry mass, and so on. Then an emergent technique in biology is a Brillouin microscopy, which means that the interaction between light and the sound waves generated by the thermal vibration inside the cells. So the interaction induces a shift at the end now in the laser, probing laser, which a very small shift, which is Brillouin shift actually, and this is measured and allows to calculate the longitudinal modulus. And last but not least, there are the laser scissors and tweezers. I report here, this image with the fiber cells. I'm a bit tension because Professor Burns looks to me when I present, but I hope to tell it well, to say it well. So in 81, in this paper, you reported the cut of fiber stress and the resealing after 1 hour. So the cut with a pulse laser. And I thought that that was the first operation, surgery. But then I learned that it was not the first. And I love very much this schematic picture in which laser scissors and tweezers are involved to keep a cell, the 2 optical tweezers, and the 2 laser scissors to operate on the cell, now to delete, to deplete a faulty gene and to open a window to introduce a sequence gene. So it is wonderful. One day this will be done for sure. And then I recently discovered this review is very, very beautiful. It's the history of the laser scissors and tweezers. So it is not because I am presenting at the BLI, but is really a view. Okay, thank you. Coming back to the methods, so we said that the estimated values of mechanical parameters depend on the experimental methods and or on the theoretical models, in addition to their dependence on cell type, cell state, and experimental conditions. So the question is, how much relevant is the measurement of a mechanical parameter for one type of cell in a certain state using a specific experimental method? Now, the point is, we as physicists or biophysicists, we think, okay, we develop a method to measure some mechanical properties for the cells. And we work years, and then we learns that maybe it is not relevant because the cell can be also like that. So biology is very, very diverse, and this should be taken into consideration. So I will give as an example the cell stiffness estimation for breast cancer cell model. So we used the breast cancer cell model composed by three cell lines in which we have myoepithelial, luminal, and basal breast cancer cells, and they are they have an increased metastatic potential. So in this sense, it is the model. So the high metastatic potential is attributed to MDA and the cells. And we wanted to measure the cell stiffness in order to, establish if the cell stiffness can be correlated to the cell metastatic potential. We used two methods, AFM and optical tweezers. Actually, just because I was wondering why, reading many papers from AFM experiments, I found for the same cell, different young modulus. And when one passes to optical tweezers measurement, very much different values for the same cell, for the young module. So the two approaches are similar, indentation approach, in which you derive the force indentation curve using the Hertz model. You derive the young modulus using the force indentation curve and the Hertz model, which is a long discussion also here about the Hertz model, if it can be truly applied to cells or not. But let us say that if all apply that, you have a way to compare with that. The difference between the two approaches, two techniques, consists in the stiffness of the AFM with respect to optical tweezers and in the loading rate. So how fast we apply the force to the probe, to the cell. Another question is where to measure for a good reliability. Because if I measure in the center of the cell, I obtain a cell stiffness. If I measure in another region, I obtain a different value. And if I want to compare with other people, I am in trouble. So we run the experiments measuring in above the nucleus and then towards the cell periphery. And what we found, both with AFM and optical tweezer, was that the young modulus decreases from nucleus to cell periphery. And since we can detect the nucleus, we can see the nucleus quite easy, we said, okay, the nucleus is defined easily topographically. So it is good that we use that region as a reference. Excuse me, my mouse is too sensitive. So comparing optical tweezers with AFM, we found that the high metastatic potential cells are significantly softer than the other two cell types. And important, the result is confirmed both by optical tweezers and AFM techniques, even if the absolute values are very different, because parameters as force range and the loading rate are very different for optical tweezers and AFM indentation. So then another question is the cell stiffness a cell type constant or it changes, adapts to environment conditions? Now I know very well this. When we did these experiments, we were not sure, and we didn't read very well the papers published in 80s. In any case, we proved that by culturing the cells to get in contact, so to see if the cell stiffness changes when the cells are in contact, and modifying the stiffness substrate. So you see here cell-cell contact, and you see the results of the elastic modulus for cell-cell contact and isolated cell. And actually what we get, is that MDA-MB cells get stiffer when in contact, looking similar to HBl and MCF is here. So the stiffness changes just because of the contact of cell-cell contact. But for CF7 and HBl100, they become softer. For the substrate, I report here only the result for MDA. What we get is that the stiffness of the substrate changes the cell stiffness very much. And again, when also the contact between cells is present, this stiffness is increased. So conclusions from this experiment are cell stiffness does depend on the microenvironment conditions, extracellular matrix cell and cell-cell interaction. And the stiffness of MDA-MB, the high metastatic potential cell, changes much more than the stiffness of the other two cell lines, which is expected, no? Because these cells have to travel, have to exit primary tumor and enter the blood circuit and then exit and fix to another organ and develop a second tumor. So they have to support a lot of mechanical deformations. No? They should be very, very flexible. So what we learned that experimental results should be always considered in the context of the experimental method and conditions, that the results validation for different experimental techniques is recommended, and comparing different types of cells or cells in different states is more reliable than measuring cells of only one type. Now I will pass to another project that I love very much on biomechanics of neurons. And this project began about 15 years ago with Professor Torre from the International School of Advanced Studies in Trieste. He is a physicist, but he is a neurobiologist, actually. He was excited about optical tweezers when visited my lab. And then he said, oh, Dan, let us try to measure the force exerted by neurons, very young neurons under development, young neurons before the network is formed. And they are very interesting, they grow and so on. And we developed a platform to do that. So I show you a movie here, a video, in which we can see the interaction between two such neurons, actually between the growth cones of these neurons. The growth cones are terminations that the neurite. So at this moment, you do not know if it is an axon or dendrite. And these axons are like, you see, my palm and fingers exploring the environment. This movie lasts 2 hours. And you see what happens is that philopodia of the two growth cones, they touch each other. And maybe, hopefully, we didn't check here, some real contacts or useful contacts were developed, and these contacts can be transformed in synapses and make the network grow. Okay? So why this interaction? This interaction is because the two cells, they release molecular cues for recognition, but also mechanical contacts probably has its role. So to measure the forces exerted by lamellopodia and philopodia, we use this approach. We placed a bit, an optical tract fixed near a cell and waited for the interaction of filopodia or lamellopodia with the bead, measure the displacement and the force. Here you can see the interaction between the lamellopodia and the bead, and the force increases at the moment, so it pushes with a force that we could measure 25 piconewton. But sometimes it happens that the lamellopodia retracted and took another direction. For instance, here is what happens with filopodia. You see, it senses a bit, it touches, and then takes a decision. Sometimes making a joke, I used to say that this neuron, a single neuron, sometimes is cleverer than a person. Because it doesn't push with all the force which is can be generated by polymerization of the actin filament. It pushes with very low forces and with touching by steps, and then taking a decision. And here is a protrusion forces, are the protrusion forces now exerted by filopodia when it's increasing. So after 10 years and so, meanwhile, we did many other projects together, but in 2015, we decided to investigate if mechanotransduction with the piconewton forces we measured for filopodia and lamellopodia is possible for neurons. And we were inspired by two works, very nice works, at least two works, I saw authors from BLI as well. This work reported in Nature, where the SARC activation by a bead stuck on the Huvic cells is moved by a laser tweezers to induce the force, and then by using fret microscopy, SARC activation is put in. But the forces in that case were lateral and were estimated about 300 piconewton. And the more recent paper by Kim et al. Reporting actually the calcium signaling pathway triggering by forces in the same with the same procedure. What we did was to use a different approach in which we move vertically the bead using a focus tunable lens, which is a fluidic lens with a membrane that can be the curvature can be changed, and you change the focal length, and this can be done very precisely and very quickly. Here is a movie to see how a bead now is displaced from the cover slip up and down, while this remains always in that position. So the idea was to use this device to create an oscillatory optical trap and to apply mechanical stimuli at piconewton levels and see if this triggers the cell calcium signaling. We used first mouse neuroblastoma cells. So we can measure the force and we can measure the indentation, which means we can measure also the elastic properties locally. The setup is shown here. So it's an inverted microscope, bright field illumination, fluorescence excitation, Hamamatsu camera with two detectors, and we can see simultaneously bright field and fluorescence imaging, and the trapping laser passing through the focus tunable lens to move the bead up and down, and the quadrant photodetector to can measure the displacement of the bead, so to measure the force. And here is an example of calcium transients evoked by piconewton mechanical stimulation. At the left, you see the cell, and here is a microbead which is not in focus, is up from the cell. And here you will see the calcium signaling when the bead pushes the cell. Okay, and again, when you saw in focus, it means that the bead touched the cell and pushed, and immediately we measured the calcium fluorescence. Okay. So then we saw that the calcium increase was noticed immediately after the stimulation. Still, here it is a bit of debate because how much immediately it means it depends on the temporal resolution of the video imaging. But then we ran also experiments with electrophysiology, so stimulation with optical tweezers and measurement for the whole cell with electrophysiology, and saw that the calcium triggering was immediately at milliseconds after the stimulation. So we can use one or multiple pulses. Here is one pulse in blue. Here is with two pulses. And we can apply different forces, changing the trap stiffness, which is obtained by changing the power laser, no? And we can use easily protocols with pulses of different lengths and variable frequency to answer to study the answer of the cell in different conditions. For instance, here we report the calcium elevation also when the stiffness was reduced at the half. So, which means that we have a modulation of the cell response with the force applied in terms of calcium transient elevation. So the conclusions here are this. We show that very low levels of mechanical stimuli are able to induce a calcium intracellular response in neuroblastoma. And this is quite new, I would say, because usually these experiments of mechanotransductions are done or with pipette aspiration or with AFM, where the forces are applied are much bigger, order of magnitudes bigger. So imagine that you stretch a cell with a very big force, then you do induce a mechanotransduction there. I will say that you kill almost the cell, but I am exaggerating. It's just to say that these small forces, locally applied, they induce triggering of calcium pathway. Recently, in this paper, they confirmed very small forces can induce extreme axon growth using multiple magnetic nanoparticles. In this case, it's not a single particle, there are multiple, but the forces are at the same level. Then the second is that piconewton forces can produce cell membrane indentation. Okay, but if we estimate the tension produced by these forces, one obtains much smaller values for the membrane tension than those previously assumed to be necessary for the opening of the mechanosensitive channels in membrane tension model or in tether models. We do not have an explication why this happens. I mean, low tension and still an effect is observed, but the fact is that the values are much lower. Piezo 1, we checked that Piezo 1 is expressed in these cells, and it's the inhibition actually of all the mechanosensitive channels by GSMPX peptide abolishes calcium transients. The cell, then we saw that the cell response adapts to repetitive stimulation and force values, and that prolonged mechanical stimulations induces the reconfiguration of the cytoskeleton. Actually, we continued the research with hippocampal neurons from mouse, and preliminary results are, so we studied the activation of the calcium transients, the activation of the calcium calmodule independent protein kinase and small G protein rho A. We saw that there is a cytoskeleton rearrangement, and the time delay between this pathway triggering is of the order of seconds and tenths of seconds, as it is illustrated here. I cannot enter too much in detail here because this paper is under review, and okay, so another experiment with the same approach we did to show that mechanosensitivity is present in the phototransduction of rod cells. And what we did was to stimulate. So this is a rod, and the rod cell has two parts: the outer segment and the inner segment. No? And we applied forces in the way that I described before on the inner segment and then on the outer segment. And we observed that calcium response is present, as it is illustrated here. So the force pulse and then the calcium increase. What we observed was that the response is weaker in outer segment than in inner segment, as is indicated also here. You see the scales. And when we checked, when we checked for the expression of mechanosensitive channels in outer segment and inner segment, we found that only in inner segment we had this expression, and blocking, again, inhibiting these mechanosensitive channels, we abolished the calcium transients. But in the outer segment, you do not have, or at least we didn't see mechanosensitive channels, and we still have a calcium increase. Why? We do not know exactly. We say that probably it is because the lipidic discs which compose the outer segment, they are deformed and they contain also the calcium. So probably there are displacements of the concentration of the calcium. Actually, we do not know exactly. The fact is that it's not due to the mechanosensitive channels in outer segment. Then what we did was to put in evidence the fact that when you flash by light a rod cell, it shrinks. So the mechanical effect on the cell induced by light. And we studied this first with video microscopy. You see here a piece of retina and the outer segments, and at the moment you should see the flash. Sorry, the flash is now, okay? You do not see the retraction by, because the retraction is very, very small, is 2 pixels there. So it's very difficult to detect by video microscopy. So we checked with single rod cell, but in that case, you do not have very physiological condition. And what we did, with optical tweezers, we placed on the tip of the outer segment, we placed a bead, and we pressed to attach it there. And then we lowered the power in infrared, and we sent a flash of light, and we measured the displacement of the bead to see the shrink. The shrinking, so the bead is attached to the cell shrinks, and you see this by following the bead, by tracking the bead. Okay, now the last part of the talk regards the biomechanics of ex vivo generated red blood cells. And this project was done in collaboration with Claudia Bernecker, who is in audience, from Clinical Department for Blood Group Serology and Transfusion Medicine, Medical University of Graz, Austria. We are neighbors, you know. and they study the possibility to generate red blood cells, blood artificially, not to obtain from donors, because there are less and less donors and more people who need blood. And this is based on the fact that ex vivo culture models are well established. Now, one needs to check the mechanical properties of red blood cells generated in this way, because for red blood cell, the mechanical property is essential. No? Is probably the most, or I don't know if the most, but it is very, very important to make its function. So the project includes the use of hematopoietic stem progenitor cells to derive culture red blood cells and to compare these cells with native red blood cells from donors and with the reticulocytes, native reticulocytes from donors, because reticulocytes in organ are the base of maturation then for the red blood cells. So a part of cell characterization requires biology, the presence of different so the hemoglobin, of proteins, and so on. Another part includes mechanics, study of red blood cells, and we use two techniques, digital holographic microscopy and optical tweezers. So to compare from a mechanical point of view, this type of cells, culture cells with native red blood cells and native reticulocytes. And well, I don't know how much time I have. I have 10 minutes more.
I think you probably can wrap up in the next 5 minutes, and we can have 5 minutes for questions, if it's okay with you.
So then I will go fast through this slide, where I wanted to say that there are many techniques which can be used to measure mechanical properties of the cells. Some of them very sophisticated, for instance, like this one, providing very nice information about the cell, but they are extremely, they have an extremely low throughput, for sure. Also, this type has a low throughput. This has, optical stretcher has a high throughput, but still we have to take into account that the red blood cell has a beacon cavity. So it is important how the cells arrives in between the two fibers to be measured. So we decided to use optical tweezers, also because a single cell trapped in an optical tweezers, if you look at the clepsydra of light formed by focusing the light, the radiation pressure mimics somehow the pressure induced by the suction of a capillary by the cell. And actually the pressures exerted by the light and the pressure exerted in micropipette aspiration for bending experiments on red blood cells are at the same level. Okay, so what was new here was, with respect to other experiments, was the use of a high-speed camera. You see here the trapping and the formation of the red blood cell with 500 frames per second, changing the power of the laser. now is 20 milliwatt, and now is 80 milliwatt. And you see the changes in the deformation of the cell. As it is shown here, now different deformations, type of deformations. And then we used also the QPD to define better the experimental protocol. So at the beginning, we let the cell like here. Then we trap the cell with a medium power. Then we change the power and we relax the deformation, and then we stretch more, and then we release, and we measure the times for cell to be deformed and for the cell to relax. And measuring this, we get that the native reticulocytes, as expected actually, are less deformable, so they need more time to be deformed in the trap and more time to recover. While the native and the CRBC are in the same range. Then we said, why not to use the signal acquired on the QPD and the power spectrum density for 1 power to see if there are differences. And indeed, there are differences. And we calculated the variance as a measure of the fluctuations of the cell membrane. And we found that the most rigid was one cell, culture cell, prepared in conditions without lipid supplements. I go a bit faster because I am not in time. Then we said, okay, let us sophisticate a bit more the type of stress. So we move the trap up and down, and this means that we move also the cell up and down, as you see here. Again, is fast acquisition. So the cell is trapped. Then we move it up. Yes, the frequency is 1 Hertz. We move it down, and you will see that it moves laterally a bit because actually we touch the substrate when we go down, in this case. So this means that we have to go higher with the trap to avoid this. So we repeated the experiments for different amplitudes of the oscillating trap. And what we found was, again, that the same cell, which was more rigid, like, or more plastic, I would say, the distance when it moves through the fluid with respect to the other cells increases. The good news was that the properties, the deformation and cell membrane fluctuation measured in this way for native red blood cell and cultured red blood cell with human platelet lipids furnished, I think I pronounced correctly. So with these two are similar. Then we said, okay, let us see if at least cell membrane fluctuation obtained with another technique gives us comparative results between cells in the same way. And we applied holographic microscopy. And this technique actually provides supplementary information very useful for red blood cell. For red blood cell, it's very important to have the shape, to have a biconcave shape. And with digital holography microscopy, you can very well measure that. You can also very nice, so you can estimate the cell sphericity of the cells and confirm that the culture cells are similar to native red blood cells. And what is seen here is that the plasma, the so-called plasma cultured red blood cells are spherical like. Also, you can determine the mean corpuscular hemoglobin, which is another important information, and the volume. No? So then we want to compare, I said, the cell membrane fluctuation, and we acquired with 110 frames per second, and we created holographic movies at this frequency, measuring the cell membrane fluctuation. It is not new. It is done also by other groups, Spark and so on. But no one compared these two types of cell. And here you see 1 second of vibration of native red blood cell and cultured red blood cell without lipids. Then from the movies, we calculate the standard deviation of the heights for each pixel, and we create this map. And then we calculate the average of this standard deviation, and this gives the CMF, the cell membrane fluctuation. And again, the results fit with what we expected, and more important, with what we obtained with optical tweezers. So the conclusions here are that cultured red blood cells, HBL, showed high similarity to native red blood cells, both as morphology and deformation under optical forces. Optical tweezers and DA-GEM show similar comparative results. Optical tweezers and DA-GEM provide also complementary results for red blood cell morphology and deformation characterization. Investigated deformation was done under fields of forces similar to those impacting red blood cells in microcirculation. And finally, the conclusions for the talk. We discussed some example of experiments investigated cell mechanics by lab. First, cell stiffness measurement by AFM and optical tweezers for breast cancer cell model. I know that AFM is not optical, but we use it as method to be compared with. And we learned some good lessons from this type of experiments. It is good that we use two methods to confirm the results. It is good to compare different type of cells instead of using one type of cell only as absolute values. And it is very good to consider the environment conditions and the experimental conditions also. Then we discussed about mechanical stimulation and mechanotransduction with piconewton forces by optical tweezers in neurons and red cells. And I think that the most important achievement here is that we show that even piconewton forces, so very tiny forces, can induce mechanotransduction in neuronal cells or rod cells or whatever. And cell deformation and membrane fluctuation of cultured red blood cells by optical tweezers and DA-GEM as a feasible tool, I would say, to prove mechanical properties of red blood cells and confirm the biological protocol now to get the cultured red blood cells. Well, this is the last in which I want to acknowledge people with who I work and help me very much, and also the people that I might miss to mention and help me very much. And thank you for your attention.
Thank you very much, Dan.